Hello, my name is Wang, Assistant Professor of Medicinal Chemistry at KU. Today, I would like to introduce our new technology to combat SARS-CoV-2. So the goal of our team is to create RNA-degrading chimeras to effectively eradicate virus from the infected cells. I have been working in the field of chemical biology and drug development for over 10 years. When I was a postdoc at Johns Hopkins University, I led the effort and developed a new type of molecular glue called reperfusions. From this new pharmacological modality, we have developed two preclinical candidates for cancer and kidney ischemia reperfusion, respectively. At KU, I've been working on a very exciting area, which is small molecule recognition of RNAs. The knowledge in my research leads to the creation of antiviral RNA-degrading chimeras described in this technology. My collaborator of this technology is Dr. Jianming Qiu at KU Medical Center. Dr. Qiu is a well-respected virologist. After the COVID-19 outbreak, Dr. Qiu's lab studied pathology in human airway epithelia and developed a specific assay, which is so far the best cellular assay for SARS-CoV-2. We have also recruited several good colleagues at KU as consultants. We meet Drs. Miao and Holmstrom on a biweekly basis for their inputs in computational biology and biophysics. COVID-19 can cause severe systemic inflammation and acute respiratory distress syndrome. We already know quite a bit about SARS-CoV-2's life cycle. So first, the infectious variant will bind to the ACE2 receptor and enter the host cell via membrane fusion or endocytosis. SARS-CoV-2 is an RNA virus. The RNA genome will be released into cytosol from endosomes. Then the translation of the viral polypeptide starts. The polypeptide, after translation, must be cleaved by proteases to assemble the viral polymerase. And the viral polymerase will then replicate the whole viral RNA genome. So currently, the drugs used in clinics all target the biological processes in this diagram. So for example, the neutralizing antibodies target the capsid protein. The protease inhibitors, such as Paxlovid, is developed to target the cleavage process. And remdesivir and molnupiravir are RNA polymerase and targeting the final replication step. It's worth noting that no agent have been developed to target the translation step right after the viral genome is released. This is the essential biological process that our technology is going after. Briefly, the RNA degrading chimera will exert their antiviral effect by reducing the functional viral transcript for translation and thereby inhibiting the whole viral life cycle. We previously summarized key RNA features in SARS-CoV-2. So for example, SARS-CoV-2 only have one single-stranded RNA in the genome in each variant. Secondly, the most mutated region for SARS-CoV-2 is located in the spike protein encoding gene. Remember, the spike protein is the target for most of the mRNA vaccines, and rapid mutation can be a problem to have an immune escape. In total, there are 271 ongoing pipelines for antiviral development targeting COVID-19. However, none of these 271 pipelines target RNA features of the virus, which makes our approach unique. What we have recently presented in the literature is a whole new pipeline for antiviral development. First, we elucidated the viral RNA structure by a type of method called chemical probing. We choose the essential and conserved sequences and structures as our target. We then identify the hits based on the structure using our in-house RNA binding compound library. Once we identify the compound, RNA binding compound, finally, we'll use a chimeric design to target and degrade the RNA structure. So briefly, an effector arm with RNA degrading activity is coupled to the RNA binding molecule. The whole chimeric compound would cause a selective degradation of the RNA target. So the degradation technology have been well documented in targeting pathogenic proteins, and this technology is named PROTAC. We are migrating the same principle into the RNA world, and this new technology has been named ReboTec in the literature. I would like to emphasize that not only we're developing drugs, but also we're developing a whole new antiviral platform. A typical drug discovery pipeline requires 3 to 10 years to progress from target validation through lead compound optimization to the selection of drug candidate for clinical trials. And therefore, if efforts are initiated only after an outbreak or pandemic occurs, it is unlikely that a very specific small molecule therapeutic will be available within a useful time frame to have a clinical impact. A potentially more effective approach is to invest drug discovery efforts aimed at building a more robust pipeline of antiviral lead structures and drug candidates. So when a new outbreak or pandemic occurs, there will be more options available for clinical deployment. Our RNA degrading chimera technology is modular and can rapidly generate lead compounds, and therefore is a perfect fit for this strategy. For the small molecule binding, we're now focusing on coumarin derivatives, which are versatile in RNA recognition. Through our past work, we now know quite a bit about the pharmacophore of this type of molecule. We discovered that the compound can selectively bind to an untranslated region of SARS-CoV-2 genome. We have performed SAR study of the coumarin derivative and obtained a nice improvement of the binding affinity. The best compound, C30, which is the red curve of this plot, can bind to the target at a dissociation constant, or KD, of 0.2 micromolar. We also validated 2 tolerable conjugation sites for the chimera design and demonstrated that the binding is selective within the SARS-CoV-2 genome. We also have 2 RNase recruiting scaffolds and a robust chemistry for conjugation reaction. And finally, we have developed a couple of cellular assays to validate the activity of the drug candidates. So based on the small molecule C30, we have made the first generation of ReboTec and demonstrated the activity in a cell model with the EC50 within the nanomolar range. Briefly, 70% of the viral genome can be reduced in merely 24 hours. Along this pipeline, we will continue to optimize and profile the molecules for finalizing the preclinical candidate selection. Very recently, we have developed the second generation of ReboTec, which is more potent than the first generation. Now the maximum RNA degradation can reach over 85%. In summary, we invented a new set of compound with the unique mechanism to combat SARS-CoV-2. The drug will eradicate the viral RNA from the infected cells. We have nearly completed the lead structure optimization of the first generation of RebelTech and are now developing the second generation of RebelTech. At this critical stage, we need resources such as investment and expertise on late-stage drug development and FDA regulations. We're also looking for partners for licensing or forming startup companies. We will file 2 provisional patent applications for 2 generations of RebelTechs, respectively. We have already attracted several fundings internally at KU and from federal agencies, especially 1/4 of a $1.2 million federal grants will be used to develop RebelTechs in the next few years. If you are interested in this technology, please contact Dr. Azrini Betha, the interim director of KU CTC, for more options and discussions. Thank you for your attention.